So today, the topic is about mitosis. Mitosis is a process of cell division, okay, of multiplication of cells. So first, we're going to talk about mitosis, and in the next lecture, I'm going to talk about meiosis. Cell division is a process that is essential for the reproduction of new cells and for you to grow or for you to repair tissue. For example, if you have a cut, you need to replace that cut by new cells. So you do cell division to repair that cut and cover it again with new cells. And another important reason for cell division is for sexual reproduction, so to produce the gametes. For us to do cell division, we have three main processes. The first one is interphase. Interphase actually is the phase of preparation for cell division. So we prepare the cells before it divides. After we prepare it, we can perform mitosis. Mitosis is when we multiply cells for us to grow or for tissue repair. And meiosis is when we produce gametes for us to perform sexual reproduction. And interphase happens in both of them, both for mitosis or meiosis. So interphase is the preparation for all of them. Okay, so interphase, we do interphase before mitosis and also interphase before meiosis. So today I'm going to explore a little bit more about interphase and mitosis. Before I start telling you about mitosis, I need to teach you one concept that is the name of diploid. Diploid comes from Greek word means diplos, that means double. What's that? Mitosis is the process of formation of diploid cells. Okay. In our body, we have two types of cells, haploid cells and diploid cells. Most of the cells are diploid. Why? Because we have pairs of chromosomes. In the human species, we have 23 pairs. In this example here, we illustrate just 3. So when we have pairs, so 2 pairs or 3 pairs, we say that cell is diploid. It has double number of chromosomes or 1 pair. 1, 2, 3 pairs in this case. When we have pairs, we say it is diploid. Where these pairs come from? From the mom and the daddy. So when we are formed, you receive half of these chromosomes from your mom and half from your daddy. When both of them are together, we form one cell that is gonna be you with diploid number of chromosomes. So we say you have 23 pairs. In this example, 3 pairs. But which cells formed you? The haploid cells. So haploid cells is the cells that you received from your daddy and your mom. And we have just one copy of each chromosome. So one copy of each chromosome, haploid. 2 copies, diploid. So how does that work? So from your mom, she has the egg with any numbers of chromosomes, just half. She does. In the eggs, there is no pairs of chromosomes, just one single chromosome for each. Your daddy is the same. For the sperm, you also have any numbers of chromosomes or just single chromosomes, not pairs. When both of them go together, forms one diploid cells. Okay, so with both N plus N, 2N. So then you form a zygote, okay, with 2N. And from here, the cells multiply and forms the whole body of the baby or the child and the adults as well. Okay, so haploid cells, just one copy of each chromosome, just one copy. And diploid cells, 2 copies or 2 one pair for each chromosome. So as I said, mitosis forms diploid cells, so 2N. So when we are talking about mitosis, I'm just talking about cells that have the pairs of chromosomes. Here is an overview, okay, but I'm going to explore each of these phases separated. So interphase is the phase of preparation after we start the mitosis itself, and we have different cycles. The prophase, the later prophase or metaprophase, metaphase, anaphase, telophase, and cytokinesis. Okay, so I'm going to talk about each of them. And here you can see illustrated the chromosomes. Let's say about the number of chromosomes. In the interphase, your cell is any cell in your body. So how many chromosomes do we have in your cell? 2N. In the interphase, what happens? This number double. So instead of you have 23 pairs, you're going to have 23 pairs times 2. So you duplicate your number of chromosomes. So when we start the interphase, you have 2N. So when we start, you have 2N. When we finish the interphase, the number of chromosomes is 4N. So in the human bodies, for example, you start with 23 pairs and we finish with 46 pairs. So you have 4N number. When you go to prophase, this number remains the same. It's gonna just condensate, so 4N. Later prophase, the same, that nothing happens. Metaphase, the chromosomes just go to the middle, but the number doesn't change. In anaphase, they start to separate it. Okay, so each chromatid goes to one side, but you have the full number yet. Telophase, we start the separation. And after the cytokinesis, you create 2 new cells with 2N each of them. So we start with just one cell. After the process, you have 2 times the number of cells. Okay, so we start with 2N and finish with 2N. But in this case, we start with 1. Now I produced 2. So I produced 1 and 2 cells. So again, in the interphase, we start with 1 cell as 2N. After here in the interphase, we duplicate the number of chromosomes. So we have 4N number of chromosomes. After we go to prophase, the only what happens here is that the chromosomes create this form. The DNA creates this form of chromosomes, so condensate, but continues having the 4N. After it starts to move, continues to have 4N. In metaphase, goes to the center of the cells, so here in the center of the cell. In anaphase, start to separate, so start to separate the chromosomes, the chromatids separate. And in telophase, we have again the nuclear membrane. And just in cytokinesis, I separate 2 cells, and now I have 2 cells with 2N. So now I'm gonna talk each of them very detailed, okay? So interphase is the phase of preparation. So we prepare the cell for the cell division. And it's not so simple. Actually, it's very long. And we have 3 phases inside the interphase. The first one is G1. What happens to the G1? You duplicate organelles and cytoplasmic components. So all the organelles, we get duplicate. So for example, you have 10 mitochondria, now you're going to have 20 mitochondria and so on. After the duplication of the organelles in the cytoplasm, you go duplicate the DNA, so the genetic material. So in the S phase, you duplicate, you say DNA synthesis. Synthesis in biology is the same as duplication or replication. So if you listen any of these three terms, is the same. So duplication, replication, or synthesis is the same. And after you duplicate the DNA, you have the G2, and the G2 you just produce the enzymes to start the mitosis. Here, when we start the interphase, what's the number of the chromosomes that we have? Yes, the number of the chromosomes of the cell. So 2n. So in the end of G1, you still have 2n. After that, what happened? Duplicate the DNA. So if you duplicate the DNA, what's going to happen to that 2n? It's going to multiplicate as well. So we have 4n now. Okay, so for synthesis, when you do synthesis, you duplicate the DNA. And in G2, what's going to be the number? 4n as well. Okay, so when we finish the interphase, we have the double number of the chromosomes. Here, I'm going to illustrate with just 2 chromosomes. So let's say one from daddy, one from mom. So in G1, you just duplicate organelles and cytoplasmic components, so you don't change the number of chromosomes yet. But what happens in synthesis? Synthesis is duplication or replication of DNA. So we had here just 1, 2, right? Now I duplicate. Now I have 2 of each. So one here and another one here. So I duplicate them. And after the last one, G2. What happened in G2? You just produce enzymes. So interphase has 3 phases, G1 as G2. G1, duplication just of organelles and cytoplasmic membrane components. S, just the replication of DNA. And G2, production of enzymes. So organelles and cytoplasm, DNA, and enzymes. It was 2n and S, how is going to be? 4n, and in G2 continues 4n. Now I'm going to explore a little bit more what's this duplication or synthesis or replication of DNA. What happens actually? So we talked about it before. So DNA is a double helix, right? Made of what? Remember? Nitrogen base, A, T, G, C, remember? So each of these here represents one base. So each of these is 1A or 1T or 1G and so on. For me to duplicate the DNA, what I need to do? First, I need to open this double helix. So I need to unwind. So I need to open this. So I need to open. So I'll go here and I open. After I open, I have 2 strands. So the one here, one in the right, and one to the left. So I have 2 strands when I open. And each of these strands is going to be a template to build a new strand. So when we open, new bases is going to come in according to the rule. So here, for example, if you have A, which base is going to come here? Yes, T. Here, if I have G, what's going to come? C, and so on. So the base, I'm going to continue according to the rule. A with T and G with C. After I finish, I'm gonna form two helix completely identical. So the two new strands is gonna be completely identical. So here illustration for you. So here is already open. When it's open, new bases are being put in, okay, according to the rule A and T, G and C, until it complete all both sides. So open and complete with the new base. Okay, when I complete all the base, I form two new strands, completely identical to the first one. Now I'm gonna talk about the mitosis process and each phase. I'm gonna start with prophase. In prophase, all the chromosome, all the DNA is gonna condensate. What does that mean? It means it was all disorganized, now it condensates. And when it condensates, it shows this format of a chromosome, of an X. Also, in the prophase, the nuclear membrane is gonna disappear. So this nuclear membrane here is gonna go away. Okay, so this go away. In the metaphase, the chromosomes goes to the middle of the cell. So goes to the center of the cell. So M for middle goes to the center of the cell. So aligns in the center. In the anaphase, ana means separation. The chromatids, so each part of the chromosome separated into two. So each of them goes to one side. So separation, anaphase is the separation of chromatids. After, we have the telophase. In the telophase is the opposite of prophase. If in prophase I condensate, in telophase I'm gonna decondensate. And here, if my nuclear membrane disappeared, now it's gonna formate again a nuclear membrane. Okay, so telophase is basically the opposite of prophase. So in mitosis, prophase, condensation of the DNA, the nuclear membrane disappears. Metaphase, the chromosomes goes to the center, to the middle. Anaphase, we separate the chromatids. And telophase, the opposite of prophase, formation of the nuclear membrane again, the condensation of chromosomes, and one more thing, the organelles and the cytosol that have been duplicated before is going to be split. So each of the daughter cells is going to receive the same number of organelles and cytosol. So prophase, my DNA was already duplicated. So I have 4N DNA. In metaphase, did I change anything? No, so continues to be 4N. I have duplicate DNAs. Anaphase, nothing changed, 4N. Telophase, now I'm separating in two. So the 4N that I have before, they go, each of them for, half of them for each daughter cell. So each daughter cell is gonna be 2N. So in telophase, I start to formate two new cells with 2N each. I'm going to explain a little bit more about what is this DNA condensation. DNA condensation is when my DNA, that it was like all double helix here disorganized inside the nucleus, it organized with the help of proteins. What's the name of the proteins? Histone. So this histone here, so this protein here, helps the DNA to coil. Okay, so the DNA coils with the help of the histone. After it coils a lot, it forms the chromosome. So it forms this structure, rod-like, called chromosomes. So DNA coils with histone. In metaphase, after it's condensate, what happens? Goes to the center. So chromosomes align at the center. Anaphase, separation of chromatids. So here we're going to explore what's the difference between chromosome and chromatid. Chromosome, we have two chromosomes here. We call homologous chromosome. Why homologous chromosome? Because one of them comes from daddy, so from father. Say the blue one comes from father, and the red one comes from mother. Okay, so father plus mother, homologous chromosomes, because they have the same genes, the same genetic information. Okay, so if here is the gene for Telophase, for example, they're gonna be in the same location in the chromosomes, the homologous chromosome, even though the characteristics are different. Let's say for daddy is the blue eyes and for mom is the black eyes. Doesn't matter. They have the same gene, and the gene may have different characteristics. But when we duplicate each of them, so here I had one A capital letter. Now I duplicate. I have two. So when I have two, I say I have two sister chromatids. Sister chromatids are exactly the same chromosome, but now duplicated. And here I have two for capitals and two for small letter, small a. So what's the difference? Chromosomes comes one from daddy, one from mom. When I have the two chromosomes, I say they are homologous chromosome. When I duplicate each of them, I form chromatids, two chromatids. So the chromatids have the same, the same gene with the same characteristics. So in anaphase, we separate the chromatids, okay? So chromosomes, shaped like an X because they are made of two rods joined at the center. The center we call centromere. So form pairs, one from the mother and one from the father. And the chromatid is each rod of the chromosome. So each part of the chromosome we call chromatid. So we say we have two sister chromatids for each chromosome. What happens in telophase? We start the separation. So we form the nuclear membrane again. So the nuclear membrane form again in both sides. So we have two new nuclear membrane being formed. The condensation of chromosomes, so the chromosomes that were coiled now decondensate, and the organelles and cytosol is split between these two. Here I have a little animation. What happens next? We call cytokinesis. Cytokinesis is just the separation of the two daughter cells. So everything happens, everything duplicates, nuclear membrane formed. Now I just separate them. And then I have two new daughters completely identical to the original. Okay, let's do a summary now about interphase and mitosis all together. So interphase, what are the three phases? What's the first one of the interphase? G1. What happens in G1? Duplication of organelles and cytosol. After we have S. S stands for synthesis. Synthesis of what? DNA. So the DNA, the chromosomes do have just 2 single copies here. Now they have 1 copy for each. After S, what we have? G2. What happens in G2? Duplication of enzymes. The 3 here, G1, S, and the G2 are from interphase. After the interphase, we start the mitosis. What's the first one? First phase of mitosis? Prophase. What happens in prophase? The DNA condensates, and now I have a very nice chromosome condensate. In this case, I have 2. And my nuclear membrane disappears. After the prophase, we have metaphase. What happens in metaphase? They go to the center. So they align in the center of the cell. After the metaphase, we have anaphase. Anaphase, what happened? They split. So the chromatids go, each of them, to one side of the cell. So they separated this chromatid, the sister chromatids. After the anaphase, what happens? The telophase. And the telophase is the opposite of prophase. If in prophase I condensate, now I decondensate. So I decondensate. And my nuclear membrane forms again. So here I have 2 nuclear membranes being formed again. And after the telophase is the cytokinesis, and I form 2 new cells, and I can start over again. And that's what happens in our body. It's continue over and over and over. Let's count now the number of chromosomes. In G1, I have 2n, n, or 4n? 2n. So 1 pair. 1 from mother, 1 from father. What happens in S? How many I have? 4n, because I duplicate. In G2, what happens? Nothing. So I continue with 4n. I don't change the number. I just produce the enzymes. After G2, I enter in the prophase. Did I change the number? No. So I continue with 4n. In metaphase, did I change the number? No. So it's going to be 4n again. In anaphase, did I change the number already? They separate, but they are all in the same cell. So what's the number? 4n. Now in telophase, what's going to happen? Still I have just one cell, but it's time to split in 2. So I have 2 cells connect together. So when I separate here, I'm going to form 2 cells with 2n each, because here I start the separation. Okay, so the 4n divides in 2, split in 2, 2n in both sides. Okay, I hope this is clear enough. If you have any question, please contact me, okay? Thank you.